Going. So this is a flu virus. This is a flu particle. They're both glycoproteins. Hemagglutinin and neuraminidase are both glycoproteins that are embedded in the membrane of the virus particle. All right, embedded in the membrane. That's why they are partially in the cell, partially out of the cell. They are embedded in the membrane. All right. First one we're going to talk about hemagglutinin. So now, one cool thing I want to tell you. Does anyone remember the swine flu? I think it was last year, a year and a half ago. Does everyone remember that epidemic that happened, the swine flu? Does anyone remember what they called the swine flu, how they categorized it? Yeah, Danny.
Influenza A virus.
Right, influenza A, and there was another one. Kevin, yeah.
H1N1.
H1N1, right. So it was H1N1. Whoops, N1. Right, so this was swine flu. Okay, so basically all flus are categorized by H blank N blank. All right, there are 17 different H's. H stands for hemagglutinin. All right, so the type of hemagglutinin, we have another one here. So here is representing, this is avian flu, this is H1. I think I was looking over some research. I found that they've categorized 17 different hemagglutinins. You don't have to remember the exact numbers, but just so you know, this could be, H3N1, H4N1. Right? This is how they categorize different strains of influenza. So for right now, this is what we got. We're not working with H1N1 exactly. N stands for neuraminidase. So H is hemagglutinin, N is neuraminidase. That's how they're classifying different strains of influenza. Infection of our cells. So we have the HA receptor. It fits in. It fits in nicely. So our cells, this is considered the HA receptor. Hemagglutinin binds, and it starts a chemical reaction, all right, at the membrane of both the virus particle and the host cell particle. And in those chemical reactions allow for the virus particle to come into the cell. Once in the cell, it will recruit lysosomes. Does anyone know what the function of lysosomes are? Yeah, Brady.
They can cause the suicide of the cell.
Yeah, they can cause suicide of the cell, and they do that by destroying cell membranes. So what we'll do here, to model this, grab a couple. It'll cause lysis of this virus particle. Right, so this virus particle will break up. So then this is gone. Oh, let's not take this with us. So this portion is gone. This breaks apart. And the viral genome, viral DNA, is going to be released into our cell. All right, so now here
is RNA or DNA?
This is viral RNA. All right, so we have the fluase and we have fluase, or this is what I want to put. So fluase is this enzyme right here, right, attached to viral RNA. So, do you want me to write viral RNA, or can we get it? It's a little bit tight on here. Viral RNA. Okay. The fluase will transport itself and the viral RNA through the nuclear pores, through the nuclear membrane and into the nucleus that have enzymes such as fluase. Because what fluase does is it reversely transcribes that RNA. So this will grab an A, come here, and it'll put on there, right? And then for here, for A, it will grab the T, right? And it'll build this strand all the way down into its entirety. Works its way down, creating there, and then it separates the strands, does a little flip, and then works its way back up as well, creating, again, double-stranded DNA. So it works its way back up the model, right, making your double-stranded DNA. Made, oops, until now we made double-stranded DNA from the viral RNA. We have RNA polymerase, right? This is from our host cell. This is host cell machinery. So this RNA polymerase then attaches, right, splits apart the viral DNA and starts adding on our own RNA.
Is it making RNA or messenger RNA?
Messenger RNA, mRNA, that's right. RNA polymerase is taking the viral DNA, which it can't tell the difference, and making messenger RNA. Now, sorry, this is meant to be pointing at the protein. This is called that protein is called integrase. So, two things that are going to be happening. Remember, this isn't going to just stop at one messenger RNA, correct? This polymerase is going to be making multiple copies of the viral mRNA. Okay, so you're going to end up with copies of mRNA here. So, let's talk about integrase now. Integrase gets transported back into your nucleus, and what it does, it takes your viral DNA, it'll open it up. Hang on the side. Okay. Thank you. It'll open it up and it'll do the same on this strand. It opens it up to allow, we're good, to allow for the integration, that's where the name comes from, to allow for the integration of the virus DNA into your genome. That virus leaves portions of its DNA in us, and then they have accumulated to the point that they believe 50% or even more of our DNA, of our genes, originated from virus particles. Right, that transports is what it does. Yeah, it's responsible for transportation. So these proteins are made, and they are going to be transported, right? They're going to be transported and embedded in your, they're going to be transported and embedded into this host cell membrane on the outside. 1, 2, those are the only ones we need right now. Also, some of the viral RNA is going to be transported out of the cell, right? And this is how we end up creating, attaches to the fluase, and this is how we create and create a new virus in a process called budding out, is the process of creating the new virus. So these migrate to the end. These start to form a bubble or a bulge in the membrane, and eventually, so the fluase RNA started to bulge, and now the neuraminidase will do this cutting action, and it'll snip off that particle. And then we've got our new flu virus that's budded out. You'll get portions of the host cell protein that has been damaged. All right, so that damaged protein is then picked up by the major histocompatibility complex, and it shows it. This is called an antigen-presenting cell. Infected, yeah, sure, we'll say that.
So whether it's a broken piece or
Yeah.
something, it's a result of the infection, the protein shows up that gets presented in an MHC.
Yes.